Hello, and welcome everyone to the fall season of the GMGI Science Hour. I'm Chris Bolzan, Executive Director, and we are thrilled to be back in your living rooms to deliver an exciting series of talks from some of the amazing scientists in our network, keeping you informed and perhaps inspired by their cutting-edge work and discoveries. We have over 100 people registered for tonight's talk, and I was so excited to see the names of both old friends and newcomers. And so for those of you who are new to the organization, I'm going to offer a brief overview and an update, and then I'll turn things over to GMGI co-founder and board member, Dr. David Walt, to introduce you to our headline speaker, Dr. Bruce Walker. GMGI was founded in 2013 with the hope that the ocean would offer a renewed source of opportunity for Cape Ann. Our ambitious mission is to address critical challenges facing our oceans, human health, and the environment through innovative scientific research and education, by bringing world-class science and transformative workforce development to Gloucester's historic waterfront. GMGI is catalyzing the local economy. And we have been encouraged by signs of this expanding local economy, even in these challenging times, with graduates of our Biotech Academy securing jobs with Lifeline Therapeutics, which opened up upstairs from our headquarters. And a new neighbor, Heffring Engineering, developer of ocean gliders for marine observation and research, announcing their plans to move in right next door. We have a beautiful state-of-the-art research institute in the heart of the working harbor, where our science team, led by our Donald G. Combs Science Director, Dr. Andrea Bodner, conducts marine research powered by genomics across 3 program areas: biomedicine and biotechnology, ecosystem function and health, fisheries, and aquaculture. Our Gloucester Biotechnology Academy, GMGI's education program, led by Dr. John Doyle, enrolled this fall its 5th class of students, who are currently training in person and safely to become biotech lab technicians in our hands-on 9-month vocational training program. We are really excited to be expanding the academy. We're going to be swinging hammers in just a few weeks so that we can double the program in size and capacity. And beginning next fall, we will include biomanufacturing in the standard curriculum. This exciting, incredible project was made possible by a nearly $1 million grant from Governor Baker's Workforce Development Cabinet and matching funds from our supporters. We're really excited about this opportunity to educate more young people from our community and to increase both their skill set and marketability with highly sought-after expertise. Though we all continue to navigate these challenging times brought by the pandemic, there is certainly no shortage of good news coming from GMGI. And we're really grateful for the donations that have been made, the support, the outreach, and the guidance from so many of you in recent months. We've received terrific industry insights on the biomanufacturing project from corporate partner Eppendorf, who also generously sponsored tonight's Science Hour. So thank you to Eppendorf for this evening. With that, I'm going to get to it. We are delighted to be bringing you Dr. Bruce Walker, the founding director of the Ragon Institute of MGH, MIT, and Harvard. And he's going to share with you insights from his work co-leading the Massachusetts Consortium for Pathogen Readiness. I encourage all of you to take advantage of this opportunity to engage with our amazing speakers, and you can use the Q&A function below to submit your questions or thoughts. I'm going to turn it now over to Dr. Walt, who will introduce Dr. Walker and also facilitate the Q&A. David?
Thank you, Chris. I'm excited to be back on the GMGI Science Hour and to introduce my friend and colleague, Dr. Bruce Walker. As Chris just mentioned, Bruce co-leads the Massachusetts Consortium for Pathogen Readiness, also called MassCPR, of which I co-chair the Diagnostics Working Group. And it was formed to focus the efforts of leading scientists to harness the immune system against COVID-19 and develop new tests, treatments, and vaccines. When not dealing with COVID-19, Bruce is the founding director of the Ragon Institute of MGH, MIT, and Harvard. He's a pioneering virus researcher, as we'll hear tonight, and an expert on viruses and the immune system. He is also professor of medicine at Harvard Medical School, professor of the practice at MIT, a Howard Hughes Medical Institute investigator, and an adjunct faculty member at the Nelson Mandela School of Medicine in South Africa. Dr. Walker's research focuses on immune responses to chronic human viral infections with a particular focus on HIV, including immunology and vaccine development. He is a world leader in the study of immune control and evasion in HIV infection and leads an international research effort to understand how some rare people who are infected with HIV but have never been treated are able to fight the virus with their immune system. And I think we may be hearing a little bit about that this evening. He co-founded the Africa Health Research Institute in 2012, which is an initiative initially funded by Howard Hughes Medical Institute to build a state-of-the-art TB and HIV research facility at the heart of these dual epidemics in South Africa. As Chris mentioned, we are very honored this evening to have Dr. Walker kick off our fall series of talks. His title is COVID-19 Learning from Patients, but I know his talk will cover a much broader landscape. Welcome, Bruce. Thank you for agreeing to speak this evening.
Well, thank you very much. And it's really my pleasure to be speaking to you. I'm sure that most of you have already been on a number of Zoom calls today, so here's yet another one, but thank you for indulging me. It was in 1981. I had just finished medical school. I had come to Mass General intending to become a practicing physician taking care of patients, a family doctor. When something quite strange happened. There was a patient transferred in when I was working in the emergency room who had been previously well, had been hospitalized for pulmonary tuberculosis. He had an acute change in mental status, and we just assumed that probably what this was extension of TB to the brain. But we got an incredible surprise as we started working him up. He had pneumocystis carinii pneumonia and Kaposi sarcoma, two lethal diseases at the same time that I had learned in medical school don't happen in 19-year-old otherwise well men. And he also had a B-cell lymphoma of the brain. So multiple different deadly diseases all at the same time. The doctor said, you'll probably never see another case like this. We have no idea what's causing this. Seems like his immune system's not working correctly. And unfortunately, within about 2 weeks, we saw another case just like it. And in fact, although it hadn't been initially recognized, it soon became clear that we were not the only ones seeing this new disease. In June of 1981, the first public report of a new immunodeficiency disease was made, but looking back, the seeds of this had been apparent for a while, just not recognized. At that time, we had no idea what caused it. We watched patients die. It was 3 years until the viral etiology was actually determined. And it took 4 years from that first case being reported before we had a diagnostic test to actually tell how big the pandemic was. And clearly it was very big. Well, I'm in the front row on the right here as having just finished my medical residency. And based on my interpretation that we as physicians were going to be the first to see new diseases as they arose, and if we didn't learn from patients, we were going to watch a lot of people die, I decided to become a physician scientist and couple my clinical skills with work in the laboratory to try and learn from those patients that I was seeing by becoming an infectious disease specialist and try and understand how the body fights back against this disease and why it usually loses the battle. So in this vein of trying to learn from patients, I want to share a clinic note that I wrote in 1994, which really completely changed the way that I saw what this disease, which up until that point I thought was a universally deadly disease. So a gentleman came in and already it was clear that something was different about him, because when I went to greet him in the waiting room, He was the only person in that waiting room in our AIDS clinic who looked well. But when he came into the exam room with me, he said he'd been infected since 1978 and related, as I wrote down here in the notes that day, that he had the sudden onset of what felt like a really severe flu. He felt like he was frozen in the mud. This happened a number of times. He called New York Hospital. He was a hemophiliac and they thought he had a cerebral hemorrhage, but in fact, that the scans were negative. He was febrile with severe headaches, had sort of viral symptoms. Those cleared over the course of about 2 weeks, and he was discharged without a diagnosis. In 1985, when the diagnostic test for HIV became available, he was tested. And David, if you remind me, I'll send you a transcript from what he described as his sort of his course through getting that diagnosis and beyond, which is really quite compelling. At any rate, when he told me he was infected in '78, I told him, well, I think they actually made a wrong diagnosis because people can't live that long. We tested him for antibodies, which tell if somebody's been infected, and in fact, they were positive. And in fact, we, at that point, had a new test that allowed us to test for virus in the bloodstream, which was always positive in people who are HIV infected. And it came back completely negative. And at that point, I said, oh my God, I cannot believe this. It means that HIV is not always a deadly disease. This person seems to be doing to HIV what the rest of us are doing to things like chickenpox and other viruses that we've had in the past. His immune system seems to be controlling this on its own. And that really changed our course, and we decided we're going to try and study these outliers. And what we did was establish something called the International HIV Controllers Study. We couldn't actually get funding for this, but through a couple in New York who heard about our efforts to try and understand how these people were achieving this incredible feat of controlling without medications, they funded us to start to gather patients that were of this same sort together. And at the same time that we were studying these outliers, we also got involved in South Africa, really at the heart of the epidemic in KwaZulu-Natal. I took this picture from the clinic where we established collaborations. 50% of the women that were coming into that clinic were HIV infected, yet did not know it. And 30% of them were transmitting HIV to their babies. There was no treatment available, even though this was a treatable disease in the US at the time. We thought if we could understand why babies do so much worse than adults, we could make real progress and hopefully figure out what was missing so that we would know kind of how to guide our vaccine development. At the same time, our collaborators there said that they really wanted to do more research. They thought that maybe we could establish a couple of small rooms at this rather worn out medical school there. And I think if we brought anything to the table, it was that we said, well, why don't we set our sights higher? Why don't we try and build a research institute right here at the heart of the epidemic? And in fact, much to everybody's surprise, over here on the right is the Dorsthoek Medical Research Institute, which was opened in 2003. You can only see a portion of it, but it was the first biomedical research institute to ever be built in an African university. And on the left here is the next vision we set for ourselves, which was, let's go beyond HIV. Let's tackle the problem of TB at the same time. We got Howard Hughes Medical Institute to invest, and now have this complex at the heart of the epidemic in KwaZulu-Natal that has facilities, everything like what we have in Boston and Cambridge. So it's really been an incredible opportunity to study the disease at the heart of the epidemic. Now, while we were doing all of this, there was an incredible setback in the HIV field in 2007 when a much heralded vaccine trial ended up being a complete bust. And that really caused us to think, you know, what can we do at this point to make progress when the things that we've tried thus far have not worked? And we thought, partly through conversations we'd had with Tony Fauci, that really what we needed to do was apply all knowledge that's available to this problem. And the way that science is set up, it's very hard to do that, because people who might be really good immunologists but had never worked on HIV couldn't work on HIV because they couldn't get funding to do it because they had no track record. We thought that there was a real opportunity here, and this was something that was really being missed. Now, serendipity comes into the story. At this point, we were working out of a mission hospital because the government would not allow us to treat patients in the public sector because they didn't believe that HIV caused AIDS. That mission hospital had an electronic medical record, and the salesman for the EMR asked me if I would meet the owner of the company to tell him that the EMR was saving lives. And I said, sure, I'd be happy to, because it was very clear to me that by this point, this hospital was treating a lot of people who were HIV infected, and they were doing a really incredible job of it because they could keep track of those patients and their symptoms and their side effects, et cetera. So that person was Terry Ragon, and I met with him for about 25 minutes, and he couldn't understand why we were trying to build research institutes in Africa when we could be using all the facilities in Boston and Cambridge. And I explained to him that there were things we could do in Africa that we couldn't possibly do in Boston, and we could get insights there, and invited him to come over if he wanted to see it. Well, that was, this is Terry meeting with one of the ancillary projects that we started, teaching life skills to kids through soccer. I handed him the microphone and asked him to give a pep talk to them, and he did an incredible job. At the end of the trip, we sat down at the airport, and he said, I'm making a lot of money and I don't want to give it all to my children because it would probably ruin their lives, but I'm sure you have all the money you need from the government, and yet I'd really like to help in some way. What could I do? And I said, well, let me explain to you how this whole system works. If we want to do a study, we have to get grant funding, which may take a year or two. And if we're trying to do something that's sort of out of the box, the grant funding agencies usually won't fund it. And so he said, well, what do you need? And I said, well, a $5,000 machine would be fantastic, that we need for the laboratory there in KwaZulu-Natal. If we had $10,000, we'd hire a nurse. If we had $20,000, we'd hire 2 nurses to do this clinical trial. If we had $100,000, we'd start a program to train junior scientists in Africa. But what I'd really like to do is try and solve this problem by bringing more knowledge to the fore to work on it. And kind of to understate it incredibly, one day he came to my office and said basically that he and his wife wanted to give us $100 million to spend over 10 years to do exactly what I had described, which was to try and bring scientists from MIT and Harvard together to work on this problem. We established the Ragon Institute. The mission was to harness the immune response to prevent and cure disease. We had the hubris to think that we could actually solve the HIV problem, so that was going to be our first goal, but we wanted our mission to be broader. Our strategy was to take advantage of the cross-disciplinary collaboration and flexible funding. And since we were embedded in what I think is the best biomedical research ecosystem that's ever been built in Cambridge and Boston, we were optimistic that we could make a difference. And in fact, I'm really excited to say that just in the past month, we've reported on a particular kind of cell. These are cytotoxic T cells that are able to release granzymes into an infected, HIV-infected cell and kill those cells. We've discovered that that there are people that actually, with their own immune systems, have been able to apparently cure themselves of HIV infection. We reported on the first 2 of these, but we now have others that we've identified, and we have a blueprint now for what we hope will be a way to cure HIV infection. Now, we may be wrong, this is a hypothesis, but we have very credible evidence that some people have been able to do this, and we have to believe that we can be smart enough to replicate that in other people. So, so here we are 40 years after the start with proof of cure, in my view, that the immune system can cure HIV infection. And in fact, we also have an HIV vaccine underway in South Africa, this Mbekodo trial with Johnson & Johnson. It's an HIV vaccine, and we expect the results from that in the next year or so, but are very optimistic that this is actually a vaccine that will work against HIV infection. Now that brings me to January this year, when I was teaching a course I teach every year with Howard Heller over on the far right in South Africa. This is a course taught to MIT and Harvard undergraduates, shown here along with some of the teachers that we invited along on this course. I think Graham Walker is one of the people listening tonight. You might recognize Sebastian Laredo there in the front row in the checkered shirt, one of the MIT faculty that was along. There were two critically important people on this trip that are shown here. Course objectives were to use HIV as a model to help students understand the many dimensions of an epidemic at a place where an epidemic was still ongoing. Student on the left is a student who just returned from visiting her family in Wuhan. She was getting text messages every day saying that something really dire was happening in Wuhan with a new pneumonia. Nobody knew really what it was. It was leading to all these hospitalizations and deaths, and was really scary. On the right is a former trainee of mine, Diana Brainerd, who happened to be the senior vice president for HIV and emerging viruses at Gilead. She was getting calls every day from people in China, physicians in China, asking her if she would release a drug that Gilead had developed to try and treat Ebola, called remdesivir, which at that point, not many people in the world had heard of, but I think most people will recognize that name. It's the first drug that was actually approved for treatment of this new pandemic, COVID-19. At any rate, the combination of these two, the combination of conversations with these two and pivoting our course so that we incorporated careful tracking of what was happening in the early stages of the epidemic with the students while we were in South Africa, led me on the way back on the airplane to think about the mission, goals, strategy, and location of what we were doing with the Ragon Institute. And it just seemed to me that there was no way we could not immediately pivot and start working on this new problem. So that's what we did. So what do we know about this disease? Well, it was a pneumonia of unknown cause, as you can see on the right. It was very different in terms of the time to recognizing the epidemic and the time to development of the etiologic agent and to diagnostic tests. It took 2 years for the first public report of HIV after clearly there were cases out in the community. This took weeks for the report to be made. It took less than a month instead of 3 to 4 years to identify the agent and come up with a diagnostic test. So really an incredible salute to the scientific community and to scientific advances that have been made that this could be accomplished so quickly. So the virus itself is what's called a beta coronavirus. It's a bit of genetic material that's, in this case, RNA surrounded by kind of a sugar coating of a spike protein and envelope protein. The spike protein is a target for antibodies that was known from other coronaviruses. We know now it originated in bats, probably transferred back and forth between bats and pangolins before it jumped to humans. But just like HIV that came from chimpanzees, here's another virus that arose from animal reservoirs and ended up in humans. So let me, just to make sure everybody's on the same page, let me make sure we all understand what the difference is between a bacterium and a virus and why I think viruses are probably the biggest existential threat to humanity. Bacteria, in this case E. coli, over 4,000 genes. It's capable of independent reproduction because it has all the information and metabolic machinery to be able to make new copies of itself. On the right is SARS-CoV-2, very tiny at the end of that arrow. It has but 10 genes, and it's completely incapable of reproducing independently. It has to infect a human cell in order to replicate, or an animal cell. What's amazing, if you think about it, is that the 10 genes and the information that's encoded by those 10 genes is able to basically control the 20,000 genes that make up the human genome and make the cell's genes do its bidding. So that is really instead of the cells making new cellular proteins, what they do is they start making new viral proteins so that the virus can go on and infect other cells. We know that now, I think today, it's over 38 million individuals have been infected worldwide. The case fatality rate of SARS-CoV-2 is only 0.5 to 1%, but it's very transmissible. Let's compare it for a second to the last pathogenic coronavirus, MERS. Very few cases, very high case fatality rate, meaning that 34% of people who got the infection died, but it had very low transmissibility. I'm concerned about COVID-19 and SARS-CoV-2, but I'm also really concerned about the next coronaviruses that we may encounter that conceivably could have the transmissibility of SARS-CoV-2, namely high, and the case fatality rate of MERS, also incredibly high, which I think you'll agree would be an existential threat to life as we know it. How does COVID or SARS-CoV-2 cause lung disease? Well, it turns out that the lungs have these receptors called ACE2 receptors that the spike protein on the virus can bind to, and that allows them to gain entry. And that's why the lung is the major site. Now, there are also other cells that can become infected, including cells in the nose, cells in the lung that I just mentioned, but also in the gut, in the circulation that causes coagulopathy or clotting, in the heart that can cause heart failure, in the central nervous system that can cause mental status disease. And there's just a report this week of SARS-CoV-2 being associated with blindness. So the infection happens, and then the body tries to fight back. So one of the ways the body fights back is through antibody secreting B cells. These are one of the white cells, the immune cells in the body. When a subset of those cells that's pre-programmed to at least minimally recognize this virus, when they get stimulated, they start to make lots of antibodies, which go through a process of rapid evolution to become very able to adhere even more strongly to the virus and neutralize it. At the same time, there's another arm of the immune response, which is infected cells being targeted by these killer cells that I mentioned to you before, which recognize an infected cell, deliver a lethal hit, and kill that infected cell. So what do we know about SARS-CoV-2? Well, you may have heard about this hyperinflammation that occurs. Let's just make sure that everybody understands. Infection is actually the virus itself causing disease in this case, and causing a reaction in the host. Inflammation, on the other hand, is the body's process of fighting against things that harms it, such as infections, by releasing chemicals called cytokines that call the immune response into action and kind of indiscriminately do that, so that you get this flood of immune cells coming in. And what that does is that leads to the release of these cytokines and chemokines that then work their way into wherever the virus is. And what you see on the next slide is what the result of that is. On the left is the normal lung air sacs with this very thin membrane so that oxygen can get into the bloodstream. On the right is something far different, and that's the lung with COVID-19, where all these inflammatory cells have infiltrated. Now that membrane is really thick and it impairs oxygen exchange, and that's really what causes the pneumonia. So we're in a situation where in some ways the immune system's friend because it's trying to fight off the infection, but in other ways it's foe because it's causing an overabundant, overexuberant immune response. So let me introduce another basic concept, and that is immunity following infection. So on the left, an uninfected individual, they get infected, infection spreads to their body, but they start to generate antibodies, and ideally those antibodies will get the body rid of virus. And on the right, you end up with ideally no virus left, but the antibodies left that can protect you in the future if you ever encounter the virus again. Now, the problem with SARS-CoV-2 is that the virus itself, when it starts to replicate and goes to high levels early on, it itself causes immune system damage. Now, at the same time, the body's trying to respond and fight the virus. And what the result of that is that you get this weakened antibody response that doesn't persist in many people. So let's think now about what we're trying to do with a vaccine. With a vaccine, what you're trying to do is prevent the damage that SARS-CoV-2 would cause by just showing the immune system enough information so that you can train the immune system to see the virus without ever having seen the virus itself. In this case, for example, by taking just the spike protein, we can make this recombinantly in the laboratory, and we can turn that into a vaccine that can then be administered. When that vaccine of just the spike protein, which is not a replicating virus, it's incapable of causing any damage itself, that can then elicit an antibody response, and you end up with immunity without ever having had to suffer any kind of immune system damage from the virus growing in your body. Now, ideally then, you're left with these antibodies, so if you encounter the virus, you'll be protected. And in fact, what we believe to be the case is that without immune system damage early on, when you immunize somebody, you get the antibody response now at a higher level, and that it persists longer. We're in the early phases of figuring out how long those antibody responses last, but it does look from animal model studies that at least over the short term, they protect from a second infection. Some of you may have read in the newspaper that there was a report yesterday or the day before of a person that did get a second infection. Second infection was slightly worse, and that's a concern, but the hope is that that's rare. That was somebody naturally infected. The hope is that with vaccine-induced antibodies, that those will persist much longer. There's another way to give somebody immunity, and this is something that you probably heard about related to President Trump. Here's a patient on the left who gets infected, and they make different flavors of antibodies. The green and red ones here have just by chance, this really potent ability to bind to the virus and neutralize it. You can, and what you can do is harvest the B cells that make those particular antibodies, clone those B cells so that you can make huge amounts of the antibodies produced by those B cells, infuse them into an uninfected patient, and thereby confer immunity that will protect that person from infection, or if they're already infected, will immediately start to work on their virus with the best possible antibodies that could be deployed. So, having explained a little bit about how the immune system works and what we're thinking about in terms of the whole concept of making a vaccine, let me say a few words about where we are with vaccines. There are over 200 vaccines that are in production, 43, probably more than that now, a few more than that are actually in human trials. There have been 11 of these that have made it into phase 3 trials, and those are large-scale trials where you test to see if the vaccine actually prevents new infections. There are 5, now 6 as of today, because Russia just approved another vaccine after very short and what I feel is inadequate testing. But there are no limited use vaccines approved in the United States or Europe at this point. Those are really Chinese and Russian vaccines. No vaccines have been fully approved for use yet, But there are these vaccine trials that are underway, and they use the same concept that I talked about before, just slightly different platforms. Sorry, I keep advancing too quickly. So one I think you've all heard of, because this is being made in Boston by Moderna, is an mRNA vaccine. This is taking some of the RNA from the virus, not all of it, so that there's no way this can become infectious, putting it in a lipid nanoparticle and injecting it to get the immune system, to get the RNA to make new spike protein in a person, and then get the immune system trained to recognize the spike protein to then provide protective antibodies. Another approach is with DNA, same sort of idea. Another is to take a splice of this viral genetic material and put it into another virus that's non-pathogenic, like an adenovirus. It's a common cold virus, and then get that virus to produce the spike protein in an infected person. The virus itself doesn't cause damage. The spike protein hopefully trains the immune system. Another I told you about, just the spike protein plus an adjuvant, which is sort of an immune stimulant, is another approach. And then inactivated virus is yet another approach, not being applied here in the US, but being done in China in particular. We, in our group at the Ragon Institute, and with Dan Barouch, one of the founding members of the Ragon Institute, have taken the inactivated viral vector approach with Ad26 virus, and basically using the exact same platform that was used for the HIV vaccine trial that I told you is in phase 3 trials in Africa right now. We do the same thing with SARS, but put SARS genetic material in here so that now you train the body to make antibodies to SARS. And what's just stunning, I think, not just for this vaccine, but for the others, is that, remember, we didn't even know what the cause of this disease was until early January. By the beginning of February, for this adenovirus vector, we already had vaccinations in animals. And as of September, this vaccine is already in phase 3 efficacy trials. Really, I don't think anybody would have imagined that it was possible to go that quickly, but that's actually what's happened. And it's not because of cutting corners, it's because the platforms had already been established that allowed this to be done. So there are a bunch of different vaccine companies. These are all shown here. I won't go into these in detail, but just to say that my own hope is that all of these work, because we need a lot of doses of vaccine. And the more we can do these in parallel and have lots of products, the better off I think the world will be. What are the key questions that we need answers to? Well, one is, does the vaccine have enough potency that it can protect against severe disease? They've actually modified the trials to make sure that that's part of the readout. How many vaccine doses are needed for protection? If you need one dose, that's 7 billion doses for the world. If you need 2 doses, that's 14 billion doses, plus the incredible logistical challenge of getting everybody back after 4 weeks or whatever the interval is for a second dose of vaccine. Hard to do in the US, reliably, incredibly difficult in many parts of the world. How soon can the vaccine be widely available? There's a lot of scale up going on, so I think that we will get to the point where it is widely available, but that is going to take a while, and I'll come back to that. Will the vaccine work regardless of age? Elderly and children are 2 groups that need to be targeted, but in the initial vaccine trials, they're not included. And it's a particular issue with children. We have to wait to see that it's safe in adults before we start immunizing children. So a vaccine for children is not on the near horizon. It will happen, but it's probably a year away. And that includes 12 to 18-year-olds, 6 to 12-year-olds, 2 to 4-year-olds, and 0 to 2-year-olds, which will all be tested in sequence, once safety is shown in adults. How durable is the immunity? We hope it's durable. Can safety be assured? I think if we follow the guidelines that the FDA has set up, I think we can be assured of safety. But what do we do while we wait for a vaccine? Well, one thing that we're doing is we formed the Mass CPR, and David Walt has been a big part of this. The idea here was to really bring all of the firepower in Massachusetts together under one roof, or one sort of, it's not a true roof, but a kind of an imaginary roof, to get scientists to talk together and to exchange information, to collaborate. We have over 500 scientists and clinicians from these different institutions shown here that are all working together. And for me, it's been the most amazing experience to see this community working so closely together, something I really, in my 40 years in Boston, have never seen before. And that, to me, has been just really incredibly encouraging. The other thing we can do is we can wear masks, and the experiment's already been done that shows that mask wearing works. Nurses in the ICU shown here were not able to socially distance, but they were able to wear masks and they did not become infected. In fact, became infected at a lower rate than people in the community. So I can't stress that enough, that this is not there's no question about it, masks do work. So let me make a few conclusions. Viruses are a potential existential threat to humanity. Vaccine development for COVID-19 is progressing at unprecedented speed. I think we'll get to having available vaccine by 2020, but available is in quotation marks because it won't be widely accessible, I don't think, to the person on the street until the second half of 2021. I think we need multiple successful vaccines. Masks really work, and we need to adhere to that. We need to prepare to rapidly adapt to future pandemics, and that's what David and I envision for the Mass CPR, is to lay the foundations now, not just to tackle this pandemic, but to be ready to turn on a dime the next time one of these occurs. There's still much to be done. We at the Ragon Institute intend to do a lot of that. Our vision is to expand our facilities. This is our rendition of what we hope to be our new facilities in 3 years. An Institute for Pandemic Preparedness that will sit in Kendall Square and really draw people from many different disciplines to work together. I want to thank all the members of the Ragon Institute, an incredibly dedicated group of people. I want to thank our collaborators internationally that have been so important to the works that we've done with HIV, and I'll be happy to stop there and answer any questions I can. So thank you very much.
Bruce, thank you for the fantastic talk. Very clear. I was watching the participants during the call, and we didn't lose anyone. So you kept everyone's attention. I think this is a new record in my history of Zoom calls over the last 7 months. So thank you so much. I'm sure there's a collective applause. Just two comments. One is I neglected to add to my introduction that in addition to all his scientific credentials, Bruce is an avid bicyclist. In fact, every Thursday mornings during our steering committee calls, he's sitting ready to, I guess, ride up Blue Hill or something, or in that area with his bicycle jersey. So he stays quite fit. And then the other comment is that, Bruce, the folks at GMGI would like the blueprint for your research institute so that we can put something that's similar to that on Gloucester Harbor.
Great. Happy to provide that.
Great. Let me get to the questions. There's some excellent questions coming in, and I'll be monitoring them. So please keep them coming. First question is, can you vaccinate with different vaccines against the same pathogenic virus?
Yeah, you probably can, whether you want to do that or not. I mean, for example, you could, most of the vaccines that are in development right now are directed at the B cell arm, the antibody arm of the immune response. But remember, I showed there were two different arms. There's this cytotoxic T cell or killer cell arm as well. The vaccines in development right now, most of them are not trying to develop the T cell part of it, but we think actually a B cell targeted vaccine to make antibodies and a T cell targeted vaccine to make these killer T cells would make a lot of sense to give together. And in fact, we're working on that. The idea is that T cells have a different mechanism of recognizing viruses. They can recognize internal viral proteins, which are necessarily usually more conserved. We've recently used network theory, social network theory, to examine viral protein structure and have found this amazing conservation of certain residues that are really important to the network staying together, if you will. And we believe those are really important targets. But what we've found, we haven't published this yet, but what Gaurav Gaya, a former trainee of mine and now a faculty member at the Ragon Institute, what he's found is that, in fact, you can identify areas of the virus to target that are conserved across many different strains of coronavirus. So we think this is a way to actually prepare for the next pandemic by having these T cells already there. So hopefully more about that in the future.
Okay, lots of questions coming in. So you described the threat of a future pandemic with the transmissibility of SARS-CoV-2 and the morbidity of MERS. Do you see a danger that a bad actor might be able to engineer such a pathogen?
You know, if you look where we are right now and compare that to where we were in 1960 and you draw a line, it's hard to imagine that somebody couldn't do that. Sadly, I think the overwhelming data is that that's not what's happened here, but that's why I think these recombinant gene committees are really critical. And, you know, I think that, you know, science has the ability to do an incredible amount of good, but we also need to be clear that we keep the focus on the doing good part of things.
The next question comes from Tim O'Donnell, one of the GMGI employees. Although there have been only been a limited number of reinfections, is there anything we can learn from those patients who have contracted the disease twice?
Yes, there's a lot to be learned. The problem is, for example, with this Nevada case that was just reported in Lancet this week, the problem was that there's an incomplete data set from his first infection and the follow-up of his first infection. So we don't really know what kind of immunity had been generated by his first infection. We have the sequence of the virus, but we don't have plasma to know whether he actually generated antibodies. So this is going to take careful follow-up. And there's a paper in Nature this week looking at other coronaviruses and how often, these are the less pathogenic human coronaviruses, looking at how often one sees reinfection. And reinfections in those cases occur, you know, 6 to 12 months after the first infection. And the data are pretty clear. Now, is there reason to think that SARS-CoV-2 is going to be different than those? Sadly, I don't think so. So I think that the immunity is going to be short-lived. Shiv Pillai, one of the Ragon investigators, did a study looking at people in the early acute stages of infection and showed actually that the place where antibodies are generated and really learn how to become even more effective is within lymph nodes. The virus itself destroys those little germinal center factories that help to mature the antibodies. So there's very clear evidence that there's damage being done. And so I think natural immunity is likely to be short-lived. And I think that the thought that we could just sort of say, oh, what the hell, let's just go for herd immunity, I think is really ill-conceived, because we have no evidence that the immunity that you get from that is going to last. Now, if you take the historical data from the other coronaviruses, and you say 6 to 12 months after infection, they can get reinfected, then we haven't really tested that in this infection because we're only 6 or 7 months into it. So we haven't begun to see that yet. So it would be really, I think, a terrible idea to just say, oh, let everybody get infected.
Okay, I know we're jumping around a bit, but Bonnie Fendrock asks, if babies and young children do not appear to get very sick or sick at all from COVID-19, why do we need a vaccine for that population?
Because they can transmit. And so, yeah, you don't need to worry about them getting sick, although some of them do get sick and some of them die. So it's at a much smaller level, and we have to, as a society, make decisions about what we tolerate in terms of morbidity and mortality. But the major problem there, as I see it, is that they're a vector for transmission, and that's why there's an ongoing concern about that.
Besides vaccine development efforts, what's being done in using drugs that can interfere with viral protein functions, such as preventing a spike ACE2 protein interaction?
Yeah, well, a lot. So we've done a lot of this in the Mass CPR. One of the early things that we did was try and look at small molecules and other drugs, these libraries of drug compounds, to see if any of them would have activity just by chance against critical viral proteins that in SARS-CoV-2. The major focus has been on enzymes that the virus requires for replication, because those are unique to the virus. And you have to recall from that early slide that I showed, we're now almost 40 years into the HIV pandemic. We've gotten it under control, not because we developed a vaccine, but because we developed drugs that work. And it may be that that's the way we get out of this one, is that we develop drugs that work. We do have, I think that the Regeneron and Lilly antibodies are probably going to work incredibly well. The problem is, can you make enough of those, and can you deliver them to enough people, and can you get them to people on time? Because there's probably a short window where those are going to actually be helpful. I think what the Regeneron antibodies suggest is that if we can induce those kinds of neutralizing antibodies with a vaccine, we'll have success. I think there's no question we know what we're looking for. We know also that the virus itself does not change much. Huge difference from HIV, because that's one of the major problems with HIV, is that it's just so variable, because it has an enzyme that makes mistakes as it replicates the virus, and so you get all this diversity that the immune system has to deal with. That's not the case with SARS-CoV-2. So I'm optimistic about drugs, and I think drugs could be the effective drugs would be a huge game changer if we can get there.
Great, thank you. Just a couple more questions, and then we'll turn it back to Chris. So from Nara Kum, hello, Dr. Walker. You mentioned that immune system damage occurs with COVID-19 infection. Is this a given?
Well, it's a very good question. We don't have as much data on people that have less severe disease to know the answer to that question. So in other words, if somebody has asymptomatic infection, is the same kind of damage happening in their lymph nodes? Because they're asymptomatic, they haven't come in, there haven't been procedures available to get lymph node biopsies from those individuals. So there's a lot to learn. I think that scientific field has progressed with just incredible speed in terms of creation of knowledge, but there's a lot left to do.
So here's a interesting question from Eric Huang. Will the upcoming flu season compromise clinical trials in any way?
That's a very good question. the people that are going to be susceptible to influenza are the same people that are going to be susceptible to SARS-CoV-2, and it'll be because they're not wearing masks. The advent of the pandemic actually nearly completely shut down the influenza epidemic last year. I mean, really to an extraordinary degree. So, to be able to test the vaccines, you have to have people get infected. I think there will be far less influenza this year because such a large percentage of the population hopefully will be wearing masks. But yeah, it will, it has the potential to complicate things. The vaccine efficacy is being measured by RNA tests so that we document that people actually have SARS-CoV-2 and not influenza. So I think we'll still be able to get the readouts.
Okay, last question. This is kind of big picture. There are 3 prongs to solving the pandemic: vaccine, therapeutics, and testing. Do you have any thoughts on the relative values of each?
That's a really good question. I think that we really need to do all of those things in concert. I guess I don't want to, I don't want to play, I don't want to play favorites here, I don't think at this point. I think that, that testing's important, but it's really mostly because people don't wear masks. I think if people wore masks, we would not need to do that. And if people just, did the distancing, the hand washing, and the masks, then, this enormous amount of testing would not be needed. And in some ways, it gives people maybe a false sense of security, especially if they're using the antigen tests, which are less sensitive. Antigen tests may do a better job of telling you who's most highly infectious, but they probably miss a number of people. And so, if you're trying to just sort of not wear masks and just get tested frequently, that's not a good solution.
And as someone who's, as you know, very involved in testing, testing will not end the pandemic. You could test everybody on the planet, but if they don't socially distance, wear masks, and we don't find effective therapeutics and vaccines, it doesn't matter how many tests we have. Yeah. Well, thank you, Bruce. This has been excellent. It's a great kickoff event for us for our Science Hour, and I'm now going to turn it back to Chris. I know there's some more questions. If perhaps Ashley can compile them, maybe send them off to Bruce. I'm sure you'd be happy to answer some of them. But Chris, I'll turn it back to you.
All right. Wow. Thank you so much, David and Bruce, for so generously sharing your time and your insights and your thoughts with us tonight. That was unbelievably informative. And thank you both also for your amazing work on these fronts for all of us. Thank you to Eppendorf for the sponsorship tonight, and to everyone out there for joining us. Like David said, we had a lot of staying power in this audience tonight. I've never seen anything like it either. I think it just is a real testament to the amazing information you were presenting. It was awesome. We are going to post the video on our website. All of the Science Hour talks are available on our website. You may have noticed our website got a major upgrade over the summer months, and the talk will be up there early next week. So we've been getting some questions about that. It'll be available then. And as you all saw in our previous invitation, we have a terrific lineup of speakers this fall. And the next one is going to be on November 19th with the 2019 Nobel Prize winner for medicine, Dr. Bill Kaelin of Dana-Farber. You are all welcome to share the invitation with others whom you think would enjoy listening. We had a really great range of community supporters and scientists on tonight. Lots of new names. I was really, really excited and encouraged to see. So we're going to stay in touch with you, and we'll keep sending you updates and newsletters. We really love hearing from all of you and appreciate the feedback. And we truly look forward to the day when we can all gather together again in person safely at our institute on the harbor. So keep washing those hands and wearing those masks. Good night, everyone, and thank you.
Good night. Thank you.